Today we're going to talk about the light-dependent reactions of photosynthesis. And what we're looking at here in a very simplified model is a cross-section of a membrane designating the inside and outside of a thylakoid. We recognize probably right away the phospholipid bilayer, which forms the basis of that membrane. But embedded within that membrane are two clusters of green ovals, and those green ovals represent pigments. In this case, for simplicity's sake, we're going to assume that those are a bunch of chlorophyll molecules because those are our main pigments in plants. You know now, though, that there are also other accessory pigments in a wide range of colors to maximize the absorption of energy throughout the entire visible spectrum, and we can assume that those are embedded in these clusters as well. But again, for the sake of our understanding in this basic model, we're just going to act like they're all kind of basic chlorophylls. What's interesting is that these two clusters have names that are very similar to one another. The first one we say is photosystem 2, and the second one is called photosystem 1. And that's a little confusing because traditionally 1 comes before 2. But what's happening here is that the name reflects more the history of discovering these photosystems. While photosystem 2 comes first in the membrane, it actually was discovered second compared to photosystem 1. So that's a little bit confusing. We just have to remember that photosystem 2 comes first. And then again, those photosystems we've discussed are clusters of pigments and proteins embedded within the thylakoid membrane that kind of facilitate these reactions. What we haven't really figured out is how exactly are plants able to kind of harness that solar energy and convert it into biochemical energy. And by kind of examining what's going on with the electrons in these molecules, we'll get a better idea of how this happens. So, at any given time, these cells, our plant cells, which have these chloroplasts and thylakoids in them, are exposed to sunlight. And when the sunlight hits one of these molecules, those little photons, or those packets of light energy, are going to overly excite an electron within that molecule. And so this arrow here represents an electron from this first chlorophyll molecule that was struck by that light energy and became overly excited that it's as if it jumps right out of that molecule. And so this chlorophyll molecule is now missing an electron. And what we talked about in our basic chem unit, we know that molecules aren't going to be very happy if they don't have that balance of protons and electrons. That's kind of what makes them who they are. But something else to keep in mind is that at any given time, if light is striking this chlorophyll molecule, it's likely going to also strike this one because light is kind of coming from all directions at all times. And so this chlorophyll molecule would also have an excited electron that would jump clear out of the molecule. So we have to figure out how are we going to meet the needs of these molecules that have lost an electron, both in photosystem 1 as well as in photosystem 2. Interestingly, at the end of photosystem 1, that excited electron is going to run into a carrier here that we call NADP+. And that electron carrier will couple with some free hydrogen ions that are out here, some H+, as well as those electrons, and it will become NADP just by itself. That molecule, NADPH, is going to go away to the light-independent reactions, or the Calvin cycle, which we'll talk about a little bit later. So all that's really happened so far is light energy excited an electron of chlorophyll in our photosystem. It jumped up and has been whisked away by NADP+, and it has now become NADPH, which tells us that it is carrying those electrons away to another reaction. So this guy is missing an electron still, as is this guy. This electron has been taken away, but what happens to this one? there's not actually a carrier that's going to take it away. Instead, it's going to travel through the phospholipid bilayer in a series of kind of passes, almost like one would play hot potato. There's a bunch of transmembrane proteins embedded between photosystem 1 and photosystem 2 that make up something called the electron transport chain. So this first proton embedded in this membrane will kind of act as the electron acceptor, but just like in hot potato, it's too much energy, and so it kind of passes it along. It's too hot to hold. And so it passes it along to the next one, and to the next, and to the next, until finally, at the end of the chain, it gets passed along right here to the chlorophyll molecule that was missing one. So it was initially missing an electron because it was excited and taken away by NADP+, and it was replenished by the electron from photosystem 2 that got passed through the electron transport chain. So this chlorophyll molecule is back to normal. This one, on the other hand, still needs to be addressed. Meanwhile, though, it's worth noting that as this electron gets passed down the chain, with each consecutive passing, it gives off a little bit of energy, and that's how it kind of gets lowered back to its original energy state and can return to a chlorophyll molecule. That energy is used to shuttle H+ ions or protons across the membrane through active transport. So this large protein that kind of spans the entire membrane is going to serve as a protein channel to actively pump H+ ions into the thylakoid, into this interior space. And so that energy that's harnessed from the electron transport chain has been used at this point to continually pump more H+ ions in. It's actively transported, which is why it needs that energy, and it's forcing those H+s inside the thylakoid where there's already going to be a lot of H+ ions. So it's going against the gradient. But why are there so many H+ ions inside the thylakoid? So remember, this chlorophyll needs to have its electron replaced. And the way that we do that is by splitting a molecule of water. We know water is a reactant of photosynthesis, but now we know why. If you take a molecule of water, it's made up of hydrogens and oxygens. Well, we know oxygen gas is given away as a waste product of photosynthesis, so we can kind of disregard the oxygens of water. But there's still those two hydrogens, the H2. Those two hydrogens are really just a proton and an electron. And if we strip away the electron, say to replace this missing one here, you're left with just H+s. And so each time we strip water of its electrons to replenish the lost ones in photosystem 2, we create an awful lot of H+ ions. And so between the H+ ions of splitting water and the ones that are actively pumped into the thylakoid, we build up a pretty significant H+ ion reservoir, or a proton reservoir. So it's very rich in H+ ions. Now, that's a good thing, because at this point, we need a lot of H+ ions. We had excited electrons. One jumped up here and was taken away. It was replenished through the electron transport chain. And this one that was lost has been replenished by water. So all of this work just to create these H+ ions. Why do we need that reservoir? Well, if we build up a high concentration of H+ ions, they're going to want to passively diffuse across the membrane in order to go to a region of lower concentration. And so that's what happens. They have another protein channel, and it's a very special one called ATP synthase. Remember, proteins that end in ase suffixes typically are going to be enzymes. And so this enzyme is the one that's responsible for creating ATP. And ATP is biochemical energy. So we're using the solar energy or the light energy to excite electrons, and that forces the thylakoid to create this H+ gradient, which will pass through that protein channel, ATP synthase. And as they pass through, it cranks the protein and converts potential energy into biochemical energy. So ADP molecules will be joined up with a lone phosphate group to regenerate some ATP molecules. And so the light reaction uses the light from solar energy to convert into ATP. And really the whole goal is to build up H plus ions on the interior of the thylakoid so that there's a constant supply of H plus ions to trickle through that ATP synthase. We know the cell needs that ATP to act as kind of a currency exchange of energy. It's a very spendable form of energy. And so we want to ensure that the cell has a constant supply of that by making sure that there are always going to be H plus ions to move through that protein channel.